Hi, my name is Johan Fernando, and this year I worked on a BioAI for predicting small molecule anti-infective drugs. So the first question to ask is, what is an anti-infective drug? They're an alternative to antibiotics. Those typically kill bacteria which are harmful to humans, which, although it's very effective in the short term, causes long-term problems. The biggest issue is that in removing all parts of the population which happen to be resistant to the drug, you create a population of superbacteria which are able to survive through the antibiotics. Anti-infective drugs dodge this problem because they don't kill bacteria, but instead simply make them unable to infect humans. These bacteria still reproduce and therefore maintain the population without creating resistant bacteria, but still render them harmless to humans. So anti-infective drugs will extend the effectiveness of our drugs and allow hospitals to become far more safe and effective places for treating patients. Sortase A is our specific target. It allows these bacteria to use their virulence factors to infect mammalian cells. The way we're going about this process is by trying to innovate in the drug design space. Currently, drug design happens by the physical chemical testing of thousands of drug candidates over tens of years. This process results in one potential drug, oftentimes which fails in clinical trials. Our proposed solution is using an AI to screen out candidates before they require potential testing. Drugs which are inherently too large or will not bond properly at all can be safely dismissed. This will save both physical resources and lab resources, in that less tests will have to occur, but also time and money, and allow the process to go much more quickly. This will get patients the drugs they need and ensure that hospitals are always well stocked with drugs that are not going to run out of effectiveness over time. There's a few relevant pieces of design philosophy that we wanted to maintain throughout the process. First, we were trying to avoid de novo design. Our mentors in the bioengineering department have seen an industry that it has limited effectiveness, and therefore we were trying to step away from it. There's potential ethical concerns here, and as it seems ineffective, we wanted to avoid it. Basically, de novo design is constituted by free, unrestricted design of the drugs by an AI. Instead, we sought to be as specific and targeted as we could be. Furthermore, we wanted to reuse code and data from as many previous places as we can. That's because all of us that were working on the code were fairly new to chemistry, and so we wanted to reuse the industry standard software and tools wherever possible to increase the stability and reliability of what we were constructing. This whole project was heavily connected to bioengineers and the wet lab they worked in. The target and model for our AI were both designed in previous projects, and the BioAI peers we had helped us to combine our drugs and make the most effective candidate we could. Our fundamental process for creating a drug candidate was fairly simple. We found publicly available libraries of drug candidates and tested each fragment individually to see which ones were most effective in binding to our target. We then combined and tested those combinations in order to see which ones would be the most effective. All of this was done digitally and was done prior to physical testing, which allowed us to be far faster and more efficient than the modern techniques would have been. We were able to save incredible time. The industry standard software, which is currently in use on a singular basis, and it's not able to test in bulk, was already far faster than physical wet lab testing. But our program both automated more of the process, which allowed it to be even more hands-off, and functioned more quickly than the current industry standard. There's a few different future directions that we want to take this project in. First, we want to automate the next step, which is the combination of those fragments, instead of the manual process that we had to do this year. Part of this was due to budgetary restrictions. The efficient software for doing this combination was out of our budget scope, but we believe that in future years, this project will be able to be accomplished. Also, while we targeted sortase A, the process itself is actually target agnostic. This means that future projects and applications will be able to have new targets and solve, hopefully, many other diseases and issues in hospitals and medical care. Finally, we wanted to create a GUI for our product. While currently it isn't a usable state, it runs in the command line, which means that only people with some degree of computing knowledge will be able to use it. If we create a user interface for it, then we'll be able to expand it to BioE and medical professionals who do not have experience in using the lower level functions of a computer.